Hi guys, today we'll be talking about cell-mediated immunity and what the power of T lymphocytes have. Presented by Bexy Hernandez, Diego Castillo, and Heaven Charles. Our table of contents include the functions of T cells, their activation, the activating signals, their cellular receptors and ligands, the immune reactions, CD4 subclasses, the activation of macrophages, and CD8 functions. What are the functions of T cells? T cells are key defenders of pathogens that are intracellular. They work hand in hand with many other immune cells such as macrophages and dendritic cells for our adaptive immunity. Helper T cells often secrete cytokines, stimulate macrophages, and recruit other immune cells depending on what kind of pathogen is attacking, but some examples include neutrophils and eosinophils. Cytotoxic T cells directly eliminate the pathogens, and regulatory T cells can just basically make the T cell at rest. Cytotoxic T cells are often recognizing MHC class 1, whereas CD4 recognize MHC class 2 by the antigen presenting cells. What are the steps in the activation of T lymphocytes? Initiation of the activation of the T cells begin in the peripheral lymphoid organs. The naive T cells must first recognize an antigen that is compatible with their receptors in order to be activated. Then proliferation and differentiation occurs, but no actual effector functions are performed by their naive T cells. As they go through clonal expansion, the T cells are able to divide to their role as either an effector or memory cell. Effector T cells go into circulation to find the antigen it was designed to recognize in peripheral tissues, usually guided by cytokines. Memory T cells are able to be essentially saved in the case that they are exposed to the antigen again, in which they are able to recognize and respond to it the same way an effector cell would. They usually are also in circulation or stay in the tissues. Effector cells usually die off after infection is eradicated, whereas memory cells are long-lived and remain. What signals are needed to activate T lymphocytes? The initial signal is usually the recognition and binding of an antigen. The receptor is able to recognize the MHC with its peptide antigen, while the co-receptors CD4 or CD8 recognize the actual MHC molecule. This helps stimulate the activation of the T lymphocyte through the TCR complex. Co-stimulators are the second signal to amplify the stimulation of the T lymphocyte. These are often on APC, which are known to present to CD4 T cells, but can also stimulate CD8 T cells during cross-priming. This is usually what induces cytokine secretion, which then may lead to proliferation. IL-2 is a cytokine responsible for expansion.
T lymphocytes need the interaction between cellular receptors and ligands, specifically involving antigen-presenting cells. The T cell receptor is able to recognize antigen peptides presented by MHC molecules with CD4 T cells bound to MHC class 2 and CD8 T cells interacting with MHC class 1. The co-stimulatory signals are critical for full activation with CD28 binding to B71, CD880, or B72, CD86 on APCs. Meanwhile, CTLA4 acts as an inhibitory checkpoint. LFA1, ICAM1, and CD2, LFA3 are adhesion molecules that stabilize the T cell APC interaction. Furthermore, there are cytokines receptors such as IL-2R that bind to IL-2 that drive clonal expansion and differentiation into certain T cell subsets. These interactions are what ensures an effective response while also keeping regulations to prevent any excessive activation. The types of T cell-mediated immune reactions. There's two types of reactions, cytotoxic responses and helper T responses. Cytotoxic T lymphocytes CD8 T cells directly kill infected or foreign cells that are presented on MHC class 1 molecules and release peripherin and granzymes to induce apoptosis. Helper T cells CD8 T cells regulate the immune responses by secreting cytokines and activating other immune cells. Subclasses of CD4 and their role. There's Th1 cells that promote interferon gamma to activate macrophages and enhance cytotoxic T cells responses against pathogens. Th2 cells secrete IL-4, IL-5, and IL-13, promote B-cell antibody production and eosinophil activity, helping in the defense against parasites. Th17 cells produce IL-17 and IL-22, driving inflammation and recruiting neutrophils to fight bacterial and fungal infections. TFH cells support B-cell maturation and antibody class switching in germinal centers.
What mechanisms do T cells use to activate macrophages? Classical activation involves Th1 cells, which will produce interferon gamma, a cytokine that activates macrophages. The Th1 cells will bind through the MHC class II molecule as well as co-stimulators of CD40 and CD40L in order to release interferon gamma into the macrophage, allowing for the secretion of cytokines, killing of phagocytized bacteria, and increasing the expression of MHC and co-stimulators. The alternative activation involves Th2 cells, which will secrete interleukin-4 and interleukin-13 cytokines that promote the alternative activation of macrophages. And when these macrophages are activated, they will mainly play a role in tissue repair and resolving inflammation, which are mainly a factor of wound healing and fibrosis. And here's an overview of what that looks like for both of them, where the Th1 activated macrophages will have different actions such as phagocytizing and killing of any bacteria and fungi that have been consumed by the macrophage, as well as inflammation. But inflammation is also shared by Th2 activated macrophages, which are mainly for anti-inflammatory effects, wound repair, and fibrosis. So how does CD8 plus cytotoxic leukocytes kill virus-infected cells? Well, CD8 cytotoxic leukocytes will recognize infected cells when they present viral peptides through MHC class I molecules. Once recognized, cytotoxic leukocytes will then release peripherin and granzymes. The peripherin will form pores on the cell membranes of the target cell, which will allow the granzymes to enter the cell and induce apoptosis. The extrinsic pathway also includes the ligand FAS induced by cytotoxic leukocytes, which will bind to FAS of the target cell, causing apoptosis. This would be in case the CD8 cell were not to be able to enter the target cell, and had to do it through the outside. And thank you for watching.